You don't need to take it. You're fine. You're doing science.
I get signal from University of Maine. I'm getting UMaine Wi-Fi from my house. So I'll be like, I gotta cancel Taiwan. I don't need Taiwan. UMaine signal so much faster, and like
it's working.
So we had like, I think we had like.
It should be.
Yeah. I actually get signal from UMaine because when I trace back from the IP address, you go back to UMaine IP address.
Yeah, but you probably can't like look at none.
Yeah, no, we do everything wrong. I told him a lot of things. I told him a whole lot of things. Nobody can do anything. Because my computer is not registered to a UMaine thing.
All right, let's get started. Okay, so as promised,
Y'all shut up now.
the first answer is, all right, here we go. So as promised, we have no longer dealing with carbohydrates. Yay. We're now moving into something a little bit more interesting, and that's lipids. Yeah, it is. Yeah, you kind of need them, all of your cells would just burst without them, and you'd kind of be a little bit of a puddle. So, I'd be happy for those lipids. I also study lipids in my lab, so yes, it's more interesting. Okay. We'll see it, by the end of this, let's see. It's about a week or so that we're going to spend with lipids, this lecture and then all of next week. We'll go through all of what kind of lipids there are in our bodies, as well as, of course, our cell membrane that we all need. And then we'll also talk about how we make them, how we break them, and then also some of the special ones like cholesterol, which is very important, and as well as bioactive ones like the eicosanoids. And so all very, very pertinent for pharmacology. We play around with lipids a lot with our drugs. So it's important to know what they're doing and how they're doing it. The other one announcement before we do start, though, since we had the snow day, we are one lecture behind. What I plan on doing is we have one lecture that's kind of like a standalone lecture on amino acid synthesis and degradation that was scheduled for next Friday. That's going to be an entirely online lecture. I'm going to post it next week. Just listen to it at your leisure before the exam, of course, and come see me with questions if you have any. There won't be any like poll questions because that's kind of pointless if you're doing it at home by yourself. So it should be a little bit shorter, but yes. There'll be study questions for that one, yeah. All right. So I'll also email you guys when it's posted online. It's almost ready. But all right, let's start with lipids. So we'll first talk about lipids, different kinds of lipids, and then go into cell membrane. So learning objectives, you can look at that later. Just a basic overview. Why are we concerned about lipids? So they comprise all of our cells. Lipids make a large biomass of our bodies. We use them for not only just structure, but for cell signaling, for secondary messengers. And you've seen a little bit of this before, so we're not going to talk too much about that. They're also used for energy, and of course, for storing energy. And next time, on Monday, we'll talk about how we can use these lipids for energy purposes by breaking them down through degradation processes and how they're much more rich in energy than a carbohydrate is. Then they are also used to target certain proteins to the membrane. So if you add a lipid onto a protein, it makes it very hydrophobic, and it want to be with something that is like in hydrophobicity. So it will go naturally to a membrane. Some of them act as antioxidants, such as vitamin E, many of which can be cofactors, such as vitamin A, vitamin K, and some of them can act as pigments as well. Each lipid typically has one or more of these roles. However, there's no lipid that's magically doing every single one of these roles. When we look at the types of lipids, they're classified in different classes. We have our fatty acids. These are the simplest, and we'll start there first. Many different types, some of which are dietary and some of which we have to make. Then we have our glycerol esters. These are more for storage. We, of course, will eat some of them in our diet, but they go right back into storage once they get back into our body. And then whenever they're at their time of need for energy, they will be broken down into fatty acids, and then we get energy out of them. Additionally, these glycerol esters include the phosphoglycerides, which make up our cell membranes. Then we have our sphingolipids. These are specific types of lipids, and we'll look at the structure. They look entirely different than the others. They have a lot of roles in intercellular modulation. They are involved in membrane structure, especially on neurons in part of myelin. And then also as far as membrane surface antigens, so some of which are involved in the immune system. Then we have our sterol derivatives. These are all derived of cholesterol or some other types of steroids. So these, many of which we do get from our diet. However, we can build them as well, some of which are also vitamins like vitamin D. We also use bile acids to emulsify fats and to transport them in our body, and those are also sterol derivatives. The last class are the terpenes. We're going to talk nothing about this in our class, other than this is the fifth class of these lipids. They're the vitamins used a lot in antioxidant defense or for other types of purposes like blood coagulation or for vision and things like that. So we're not going to talk too much about those. So we'll start with the simplest and then work our way more and more complex. So the simplest are the fatty acids. Sometimes they're called free fatty acids. They're just a long chain of carbons with a carboxylic acid at the very end. So the carboxylic acid is why it's called a fatty acid, and it has a very long chain. So with fatty acids, the chain of carbons can be any number of length. The way in which that we number the carbons is always going to be consistent. The carbon that is part of that carboxylic acid is always labeled the first carbon. And then the carbon after that is always given the letter alpha. We also can call it the second carbon, but it typically is considered alpha. And then the second one away from that carboxylic acid is beta. And then at that point, the Greek letters kind of stop until you get to the very last carbon, and that is given the Greek letter omega. The reason why I'm talking about all these Greek letters is that when we talk about oxidation, when we talk about breaking down these fatty acids, there are different pathways that would break the fatty acids either at the alpha carbon, the beta carbon, or the omega carbon. And so on Monday, when we talk about oxidation, it'll be good to remember what the alpha, what the beta, what the omega carbons are. So this is the classic way in which we can name and order our fatty acids. So the fatty acid itself has a carboxylic acid. Again, the pKa of this is around 4.9. It's anionic and at neutral pH. So our fatty acids in structure, they look like a long chain of carbons. So they're usually pretty linear in shape, except when you start introducing things like double bonds. Once you get double bonds in that structure between those carbons, you start to get kinks in the chain. And so those fatty acids can become bendy and not quite straight lines. This has effects on the structure of not only the fatty acid, but whatever that fatty acid is composing of. So if it's a triglyceride made of 3 fatty acids, one with all linear fatty acids without double bonds would look much different than a triglyceride composed of 3 fatty acids with double bonds in them. So in living systems, we typically will see fatty acids of around 14 to 24 carbons in length. They can be saturated or unsaturated. So when we look at names, when they're saturated, we see the name always ends with ANOIC, anoic, and the unsaturated ones are enoic with an ENOIC. So the double bonds can be found in either the cis configuration or the trans configuration. So the trans olate, which is here. So the trans, and then cis, where the carbons are on the same side of that double bond. This is the cis oleate. So naturally occurring unsaturated fatty acids, these unsaturated ones which have at least one double bond, usually will have a cis configuration. And it will induce a bend in the hydrocarbon chain, which will significantly alter the packing if you're putting it into, let's say, a fat droplet inside of a cell, or packing into forming a biological membrane. These trans fatty acids are not naturally occurring, and we actually have to synthesize them if we actually want to use them. One way in which they actually get used is in certain types of food. The trans fatty acids usually occur during the partial hydrogenation of unsaturated fats during food processing, and they are often put into foods to increase their shelf life. We can also see some trans fats that are formed with red meat consumption as well. They're correlated with high levels of the bad cholesterol, LDL, and we'll talk about the different types of cholesterol. And of course, also with cardiovascular disease. However, the exact mechanism of why trans fats and leads to this higher LDL, the exact mechanism is not really known. It's also been implicated in inflammation. So whether these trans fats can actually induce more inflammation than a cis fat would. So when we look at fatty acids and we're numbering these unsaturated ones, so these unsaturated ones are the ones with the double bonds, there is a naming system that we should become familiar with, in that we look at, first, we'll number all of the carbons from the first carboxylic acid all the way down to that omega carbon at the end. So in this example, there are 12 carbons in this chain. So we put the 12 at the very beginning of the name, then we put a colon, then we put the number of double bonds. So in this example, we have 2 double bonds, one here and one here. So then we write a delta, and then we just put the position of those double bonds, naming that first carbon of the double bond. So here, the double bond is between 5 and 6, so we put a 5. Here it's between 9 and 10, so we put 9. So to write and notate this type of fatty acid, it'd be 12 colon 2 delta 5 comma 9. And so in this way, we can draw out what this fatty acid would actually look like. So let's practice that. How would we name this fatty acid?
1819. Oh, sorry. So while they're doing that, can I ask a question?
Sure.
So, so they're naturally occurring cis formation, but what in red meat causes it to be trans? Because red meat is, of course, natural.
It is natural, yes. There's some processing of red meat. Red meat we can either eat direct from the source, or we can have processed red meat, processing.
Like,
yeah. Yeah, or like meat in a can, that kind of thing, or that kind of thing, or any other meat that has a shelf life in a box, or something that's questionable. Is this really meat?
So natural meat is
natural meat,
processed.
right?
Okay.
Yeah, mostly the processing of this meat. Yeah. Okay, so majority has 18 colon 1 delta 9. And that is the correct answer. We only have 1 double bond, and then the location is at that between the 9th and 10th carbon. So there are a lot of important fatty acids in mammals, some of which we consume, some of which we make, and they all have different structures. So the simplest is just acetic or ethanoic acid. It only has 2 carbons, 0 double bonds. And then we get more and more complex, larger and larger, such as when we get down to arachidonic acid, get about 20 carbon atoms, 4 double bonds, and much larger. So when we look at the number of double bonds, we can see you can have one double bond or you can have multiple double bonds. When we have one double bond, it is just monosaturated. When we start getting that second double bond and we increase the number of double bonds, that is a polyunsaturated fatty acid. So these polyunsaturated fatty acids sometimes are just abbreviated as PUFAs. Yep, people actually just call them PUFAs. Yep. They're found in cold water fish and in other foods in naturally high levels, and they're very good for us. Most of these cannot be synthesized in humans and have to actually be ingested. So a group of fatty acids with the omega-3 carbon having a double bond are called omega-3 fatty acids. So basically, if you have our omega carbon, there's a double bond 3 carbons away from that omega carbon, so that we call it the omega-3s. And they seem to have protective effects on cardiovascular disease. And again, like the ideas about trans fat, all the mechanisms why these are so protective are also being worked out as well.
Can these be synthesized in lab?
You can synthesize them, yes.
For consumption or?
Yeah, there are like supplements, but usually the supplements are just naturally derived, either from some type of fish oil or things like that. It's easier to sometimes just extract and to concentrate it than it is to actually, put it in the lab and make it that way. Chemical synthesis sometimes is straightforward and sometimes it's very roundabout and more costly than actually doing an extraction. So with fatty acids, there are a couple properties that are important to keep in mind. First is melting point. So the melting point of a fatty acid is related to the degree of unsaturation. So the more double bonds, the lower the melting point. So saturated fatty acids, these are the ones without double bonds. These are more solid at body temperature, whereas the unsaturated fatty acids tend to be more liquid and oily at body temperature. The melting point of a pure fatty acid is also related to the length of the chain. So the longer the chain have higher melting points. So they have larger surface area, more carbons, more surface area, and so they have a higher melting point. The other property to keep in mind when we talk about fatty acids and then how we build them into triglycerides, into membranes, is the shape. We've mentioned this before, that without those double bonds, those saturated fatty acids are pretty linear in shape. They can be compressed and packed really compactly. Whereas something that has an unsaturated fatty acid will have bends and kinks. So you can see here how that changes the shape, where we have this triglyceride, where we have these 3 fatty acids put together, and we have 2 of which are saturated, fully saturated, so they're straight lines, but then we have this unsaturated one with only one double bond, and you can see how that makes that kink in the system. So it can help define the shape in 3 dimensions for either a membrane or for some other component that this fat is made up of. Yes?
So going back to the sexy girl in the bond back there.
Yeah, the trans fat,
Yes.
the margarine, yeah.
So off this slide,
Mm-hmm.
because that's trans,
Yes.
and it's got double bonds, how do they make it cis so that it doesn't become liquid and it's still solid? You know what I mean?
Right, so those trans fat, those have a very similar structure. See how it's now not kinked?
Right.
So it does change a little bit of the chemistry as well. And remember, this is not a pure substance. There's other components going in there as well. Margarine and other things like that are quite, they're not pure. So there's a lot of things that are in it that can help solidify and keep the shelf life and, clog arteries and all that other kind of goodness.
Is that what they used to call oleo?
Mm-hmm. Yep.
Right. Thanks.
Okay. So simplest, we have our free fatty acids. These are just our fatty acids. Now let's build up in structure and in more complexity. So next we'll talk about triacylglycerols or triglycerides. The names are synonymous. So They're usually abbreviated as TAG. So basically, you have 3 fatty acids that are held together by a glycerol molecule. Glycerol is just 3 carbons, each with oxygen to connect it to the fatty acid. So glycerol is this pink backbone, and then our fatty acids are all the blue fatty acids that are linked to it. Now, when you have a triglyceride, it doesn't have to be all 3 of the exact same fatty acids. That can occur, of course. However, majority of the time they are mixed triglycerides, so they contain multiple different kinds of fatty acids within that, attached to that glycerol backbone. When fatty acids are bound to glycerol to make this triglyceride, it's virtually insoluble in water, and this is actually a mechanism for which that you can actually store our fatty acids in things like a lipid droplet inside of a cell, store it for later for oxidation purposes to get the ATP out of it. So the cells that actually store our triglycerides are our adipocytes. These are a specialized cell which has a very large oil or fat droplet within the cytoplasm. So this entire cell here, this is one adipocyte. The nucleus and all the other organelles are kind of just pushed to the periphery, and the majority of that cell is taken up by this giant lipid droplet. As triglycerides are formed, they're just packed and packed and packed further and further into these cells. So they're specialized cells which are post-differentiated. They're not going to turn into anything else. They're just fat cells, and they serve as a major energy source. They also help provide insulation. All of the fat levels inside of here will help retain a lot of the body heat that gets produced through the oxidative phosphorylation, the electron transport chain. With all of our cells, the adipocytes are located throughout our body in major areas that help retain some of that body heat. So when we look at triglycerides, they have a 3-dimensional structure. That is important because one of the carbons is a chiral carbon. Just as we talked about the chiral carbons with carbohydrates, there are chiral carbons in these triglycerides. And it's always going to be that second carbon because there's the 4 different things attached to it. And when we look at these triglycerides, it is amazing in that the enzymes that work on these triglycerides can distinguish that first from that third carbon based on the chirality of that second carbon. So even if all 3 of those fatty acids that are attached to that glyceride, that glycerol, were all the same, the enzyme used to break that triglyceride can work only on the first carbon versus the third, or vice versa. So they can actually distinguish what is that first carbon, what is that third carbon, and will actually only work on one versus the other. One of which is glycerol kinase, which will preferentially phosphorylate the third but not the first carbon. And so when we talk about the breaking of triglycerides, this will become more apparent, and that won't be for a little while. So just something to keep in mind. So a little review point before we go further. Which will not raise the melting point of a fatty acid? Okay, looks like majority of answer to stop moving. So we have converting from polyunsaturated to monosaturated for our the last choice. Part D is the majority of the class. And it is correct. I know that says A, it's just, it should say D, it's the last one. Convert, It is A.
A is the one.
A is the one. I'm sorry.
Fatty acid to omega-3 fatty
Yes, A is the right answer. There is. Oh,
And the
it's this stupid thing. There we go. Converting the saturated fatty acid to an omega-3 fatty acid. Okay, so the greater the degree of saturation, the more single carbon-carbon bonds, the higher the melting point of a fatty acid due to its increased surface area. So if you convert from a polyunsaturated to a monounsaturated, you increase the saturation by removing a double bond. The longer the hydrocarbon chains also increases the melting points due to increased surface area and some van der Waals interactions. So if you convert to an omega-3 fatty acid, it will add a double bond and thus decrease the amount of saturation in the melting point. Okay. So we started with triglycerides. Now let's move on to another complex type of lipid. These are the phospholipids. These are the important lipids that compose our membranes. So our phospholipids have a structure of a hydrophilic phosphate head, as well as a glycerol backbone and two fatty acids. So oftentimes we call these fatty acids the tail. And these are nonpolar, they're more hydrophobic, whereas the top, the phosphate, that is hydrophilic. So if we were to look at it, the phosphate is just your typical phosphate, usually bound to an alcohol. And that is on one side of the glycerol, whereas the two fatty acids are on the other side. And they will line up together so that the hydrophilic ends interact with each other versus the hydrophobic ends interacting with each other. Some important ones that we see in our cell membrane include phosphatidylethanolamine, phosphatidylserine, and phosphatidylcholine. All of them have very similar structures where you have your glycerol in the center, you have your two fatty acids, and then you have what's changing is the alcohol that the phosphate is attached to, whether it's ethanol, a serine, or a choline molecule. Something that is interesting and to note with all of these phospholipids, the actual compositions of those phosphate tails really doesn't matter. The length of the chain, they're all going to be considered the same thing. If they have phosphatidylethanolamine and they have that ethanolamine group right here, no matter what those fatty acids are, it's still phosphatidylethanolamine. Doesn't make a difference. It's really just whatever that alcohol is what's going to define what that phospholipid is. So phospholipids not only make up the cell membrane, they also act as signaling messengers, secondary messengers such as phosphatidylinositol. Its structure is here. It is a large molecule, rather intimidating to try and think about memorizing that, so we're not going to. And so phosphatidylinositol is used in cell signaling. It is usually present in the cell membrane, and then activation of the G-alpha receptors will increase the phospholipase C activity, which will then release the inositol 1,4,5 trisphosphate, so the IP3, and that will go out into the cytoplasm and will act as a secondary messenger. The rest of the molecule, the diacylglycerol, can either be used for other processes or shutting back into the membrane. So if you have just a little bit more visual, we can see our membrane is here. We have our phospholipase C. We'll take that, that IP3, the inositol trisphosphate, out of the membrane, and then it will go into different receptors, such as the IP3 receptor in the endoplasmic reticulum, and it will help regulate calcium signaling. Diacylglycerol itself can go back into and stay in the cell membrane. It also has some receptors on protein kinase C, which can actually help potentiate this reaction. So we have a cell signaling event using these lipid mediators. Next class that we'll talk about are sphingolipids. It's really just kind of fun to say. We have basically, when we looked at our triglycerides, we had our glycerol as our backbone. Sphingolipids use a molecule called sphingosine as its backbone. And it is a huge molecule, which we, again, don't memorize this. It's just, we don't need to know that. Glycerol, that's easy to remember. Sphingosine, I won't remember that. So we have our sphingosine. If we attach it to some fatty acids, we could have something like ceramide or sphingomyelin. If we add a choline onto it, sphingosine is another one. These are all very important lipids, also present in our membranes, and can be used either for structure. So things like sphingomyelin and sphingosine are components of myelin, so that it's very important for the currents along neurons, or they can be used for different signaling events as well. And that's really all we're going to say about sphingolipids there. Yeah, that's about it.
So that was quick. So what's the major point to take from that? Those two slides?
Major point is they use sphingosine as their backbone, and they're in myelin. And some of the common ones are just sphingomyelin, ceramide. That's it. Yep. So now we'll get to the more important part of the lecture, and that's the composition of the cell membrane. And so the cell membrane, of course, is a component of every single cell. Having a proper functioning membrane creates a barrier from the inside, from the outside. And we have these we also have these lipid membranes are going to help delineate the organelles as well. Things like the mitochondria have a double membrane, of course, and then the ER also has membranes. So these making space in compartments is very important for cells, for cell function. So with the cell membrane, we have the idea of the fluid mosaic model. So the membrane is kind of like a mosaic. So in art, we have a mosaic. It's all these little tiles that are connected together to form one big picture when you stand way far back. That's kind of what it's like for the cell membrane, where you have all of these little tiny pieces of lipids, and if you stand back, you see a membrane. All together, you have, zooming in, you'd have all these different components, not just lipids, but you can also see proteins and sometimes even sugars inside of it. The hydrophilic portions of the phospholipids and for the proteins that are there are going to congregate together, and they're maximally going to expose the aqueous interface. And most of the lipids and some of the proteins can actually drift across this the surface of the membrane, and so we'll look at what that looks like. And that fluidity is aided by the addition of unsaturated fatty acids. So remember, these unsaturated ones have those kinks and have those bends in their structure. So adding all of these unsaturated fatty acids into the membrane will help give it more of a fluidity so that all of these things that can drift can actually drift through. So if we look at a cell membrane, if we were to zoom in far enough, we see that this is part of the reason why it's called a mosaic is because of all the different tiny pieces that are interworking together. We have the red and yellow phospholipids composing the majority of that membrane. And of course, the more hydrophilic ends are going to be out exposed to the environment on one side, and then also exposed to the cytoplasm on the other side, and all of those hydrophobic tails are in the center of that membrane. Studded throughout that membrane, we have our membrane proteins. Now, these membrane proteins can be a variety of different things. They can be our cell receptors, they can be channels for ions, they can be different types of proteins involved in cell signaling, they can be antigens, many different functions for these types of proteins. And there's many different types of these. They can be fully integrated where they pass through the entire membrane and go into the membrane, or they can only be peripherally attached so that the majority of the protein lies on either the inside or the outside of the cell, and they're only tethered to that membrane. And then we have a lot of glycoproteins. So some of these proteins have sugar molecules attached to them. This helps aid with becoming more water soluble. And so you see these sugar molecules usually on the exterior of the membrane pointing out towards the environment. Additionally, at the very bottom, more on the cytoplasmic side, you have these cytoskeletal filaments. These are all going to be different types of cytoskeletal proteins, which help give the membrane a shape. So all of our cells have distinct shapes. Neurons look a certain way. Muscle cells all look a certain way. And it's due to those cytoskeletal filaments underneath that membrane to help give the shape and definition to those cells. So with our cell membrane, it's a bilayer, so there are 2 layers of these phospholipids. The membrane lipids are amphipathic, meaning that they have both a hydrophilic and a hydrophobic side. If we were to look at it, they're roughly rectangular in shape. So you have the polar end on either the cytoplasmic side or the exterior outside side, and then you have the hydrocarbon nonpolar sections right in the middle. So when we look at our cell membrane and our lipid bilayer, the hydrophobic interactions are the major driving force to keep the membrane together. Water molecules can be released from the hydrocarbon tails so that there's not going to be much water in here at all. It's actually going to be pushed out to either side of that membrane. And there's going to be van der Waals forces between the hydrocarbon tails and will help to facilitate very close packing. Additionally, there's going to be some electrostatic and hydrogen bonding attractions between the polar heads and with water on the outside. So all the polar ends, of course, on the exterior of that bilayer, and so they will interact with the water to help maintain that, those reactions. So we've talked about how the membrane is fluid, that we can have diffusion of proteins and of lipids throughout the membrane. It can either be a lateral or a transverse type of diffusion. So lipids can move. This is a fluid model, so they're not static. So lipids can have very high rates of lateral mobility. So here on the top, we have a lateral move where this one phospholipid here, which is the darker pink, can move rapidly all throughout that top layer that it's part of. However, a flip-flop or a transverse diffusion where instead of being on this side of the membrane, it then moves to this side, to the opposite side of that membrane. That type of move is very slow. It doesn't happen very quickly, and sometimes actually having this type of flip can actually be a cell signaling type event. So the fatty acid structure and inclusion of certain molecules like cholesterol help determine and help make this membrane fluid. So cholesterol can be added and integrated inside of membranes, and when they do, the cholesterol is put right into that non-polar region. It will not interact at all with the polar phosphate heads. It is going to only interact with those hydrocarbon fatty acids. And so the hydroxyl group inside, there's a hydroxyl groups interacts with the top of that phospholipid, and it can act as like a plasticizer, and it can broaden the melting point. So it helps to make the membrane much more fluid. Again, components of our membrane are very important. Our proteins, we have different types of proteins that can be integrated into that membrane, where they can either be integrated or they can be peripherally attached. So here we have, for example, the cytoplasm on this side and then the outside world on this side of the membrane. we can see that proteins can go and span the entire length of the membrane. They can be tethered to the membrane either directly through some type of attachment to a fatty acid. They can only go partially through a membrane, so it only goes and it interacts with one half of that lipid bilayer. And sometimes you even have proteins which don't interact with the lipids at all, they just interact with other proteins. So here we have an integral protein here, which is interacting with this blue one right here, and this blue one is only interacting with the yellow protein. It's not interacting with that phospholipid bilayer at all. So we have the many different types of choices and compositions of these membrane proteins. So typically, when we look at these types of membrane proteins, there are some trends that we can establish. The inside and the outside of these proteins, if this is one that actually spans the entire length of that bilayer, the area of the protein that interacts with that lipid bilayer, that area of the protein is going to be very hydrophobic, so that it interacts with those hydrocarbon chains of the fatty acids. Whereas the parts of the protein that are either outside or inside but not interacting with that lipid membrane, they can have all different types of polarities. You could have very polar molecules to help aid with being water soluble, especially if you're in the outside or interacting with the cytoplasm. But all of those amino acids that are located in that bilayer, they're all going to be nonpolars. When we look at the different types of integral membrane proteins, there are certain structural motifs which are used consistently, one of which is called the alpha helical alpha helical bundle. So here we have a group of alpha helices that are put together and linked through various loops and turns. If you were to look down on the membrane, it would look like this. So where the pink would be your phospholipids, looking down onto the membrane, all of those proteins in alpha helices would be here in the yellow. These help make transmembrane regions that will cross the membrane, and they're very common structures. It has distinct domains inside and outside of the membrane. And they are often used as either a receptor or some type of membrane protein that reacts, has some type of enzymatic functions inside of the cell. This is in contrast to the beta barrel. Here we see we have all these parallel and anti-parallel beta sheets that run around in a circular shape and are connected by loops. If we were to look down, the pink, of course, is our phospholipid membrane, and then we can actually see a channel. And so this is used usually with ion channels or other types of channels where whatever is being moved through the membrane will go directly through that open hole.
So there really wouldn't be anything in the center kind of overlay, right? Because you're just looking into the cell, goes to the channel, it would be open all the time?
Well, it's not open all the time because you don't want channels open all the time. Channels do get regulated. So there would be some regulation of that. So these ribbons are kind of just showing the shape. Remember, these are all amino acids and these are all proteins that have three-dimensional shapes as well. So there's going to be portions that are sticking out in all different directions with those R groups. And so the amount of surface area in between, there might be some of that pink in between, there might be very little of it, but depending on the channel, how it opens, how it closes, whether you have an open hole or you have a hole that has to be regulated to be open or closed, that's going to be determined based off of what that actual beta barrel is.
So could this be an aquaporin?
It could be an aquaporin. It could be something like a potassium channel, a chloride channel, any of those ion channels that are important for. It could be any of them. It really, this is just a structural motif. There's many ways in which you can regulate how it opens and closes. So those were all examples of integrated proteins. So they were all reside inside of that membrane. Peripheral membrane proteins do not go through the entire length of the membrane. They will have a small portion that is attached and tethered into the membrane. And they have hydrophobic amino acids that will interact with the hydrocarbons inside of that phospholipid bilayer. So these are always going to be your nonpolars, your hydrophobics, so that they can interact with those hydrocarbons. And oftentimes they can take on any shape that resembles a soluble protein because there's only one little link to that membrane. Whatever is in the cytoplasm or on the outside, in the outside extracellular space, it can take on any three-dimensional shape. They can have this covalent lipid layer anchor, and then they're found on either side of the membrane. But when you look at each specific one, it's going to be on a specific side, so that you don't have basically these, for example, if you had one specific protein, it wouldn't be on both sides of the membrane. It would only be on the cytoplasmic side, or it would only be on the extracellular side. So the membrane has to be strong, because if you had a very weak membrane, anything that would disturb it, it would just burst. And so the membrane has very little structural strength to it itself. And so the lipid bilayer can easily become ruptured. And you can have problems with that. If you burst a cell open, you see in this picture, all the cell contents come spewing out. And so the way in which you get the strength from that you need to help maintain this integrity of the membrane is through interactions with these proteins. And so they can provide structural strength as well as three-dimensional shape as well. So if we have a cross-section, if we look at just a section of our lipid membrane, we have our plasma membrane here, as well as all of these integrated proteins. And we see that underneath this layer, we have all of these structural proteins in underneath to form a nice basement membrane for these for this membrane. And then on the outside in this extracellular space, we have extracellular matrix to help provide strength as well. So the matrices on the extracellular side are usually made up of carbohydrates, some proteins, and other types of lipids, and they're anchored directly or indirectly to the plasma membrane. The cytoskeleton is on the inside, on the cytoplasmic side of the membrane, and they provide a scaffold that will extend throughout the entire cytoplasm. The cytoskeleton will help determine the shape of most cells and the specific protein structures that connect the plasma membrane and the cytoskeleton will occur. So if we had our membrane here, we can see all these different types of filaments. We have actin, we have microtubules, we have intermediate filaments. These are all just different names. They have different structures, and some of which behave differently under different conditions. If we were to stain cells with fluorescent markers, we can see that the red dye here is the plasma membrane, and that underlying all this plasma membrane, we have these filaments which are stained in green. The blue is a cell nucleus, and the green of the filaments extend to the entire shape of the cell and help give that cell its shape and structure. It's also providing a lot of strength for the cells so that they don't burst. Oh, yeah, that too. Yeah. Of course, this is all fluorescent staining. They don't look like that in real life. So summarize membranes, they form a sheet-like structure with a lipid bilayer and will consist of mostly lipid and protein. The lipids themselves are amphipathic in nature, so they have a polar and nonpolar ends. Forms a bilayer that's impermeable to polar molecules. Specific proteins mediate distinct functions in the membranes, whether they're receptors or they're channels or whatnot. The membranes are non-covalently assembled, so there's no real covalent bonds between any of the molecules there. They're all held together through other forces. The membranes are asymmetric, so that there's different faces, so whether it's the cytoplasmic side or the extracellular side, and that membranes are fluid in structure where lipids and proteins can diffuse freely, whether it's a lateral diffusion or a transversion. So that's it for today. Any questions before we go?
This isn't about this lecture specifically, but it's a question about next week.
Okay.
For the quiz on Wednesday,
Yes.
is this lecture going to be on there? Like, where's the cutoff for that?
We don't have that yet. I'm not sure.
Okay.
Yeah.
Cool. All right.
Yeah, my dad.